All right, hello all. We are going to do a pre-class lecture here. So in this little mini lecture, I'm going to talk about membrane transport as well as active and passive transport. And then we'll talk about exocytosis and endocytosis on Friday. So when we're thinking about cells, right, cells maintain these gradients between what is on the inside versus what is on the outside. Right? So cells are really good at maintaining this constant internal environment that's different from the environment around it. And one of the gradients we might say exists is a transmembrane gradient. So it's a gradient on either side of the plasma membrane. We see in this image up above here, right, that the concentration of the solute is much higher on one side of the membrane, which happens to be outside of our cell, versus inside of the membrane or inside of our cell here, right? Now this could be reversed. We could have a concentration of something higher on the inside versus the outside, right? Big picture here is that there's a gradient across a membrane. We can also have something what's called an ion electrochemical gradient, right? And this is when we start talking about charges, right? So it's a gradient that exists between molecules or solutes, just like we saw in this above picture here, but now we have the added complexity of charges. All right, so not only do we have to, the cell has to regulate what solutes are on the inside versus the outside, but it also has to regulate the relative charge from the inside to the outside. All right, so it's a gradient involving ions. Now if we think about our phospholipid barrier here, all right, we know that we have our phospholipid. It has the hydrophilic head and the hydrophobic tails. Right? So these hydrophobic tails, they're going to all align towards each other and form this bilayer. Right? Now because this forms this bilayer where one side of it, or the outside of it, will be hydrophilic and the inside is hydrophobic, right? It poses a barrier for a lot of different molecules to easily pass through this membrane, right? So we call this selective allowance of molecules across the membrane a selective permeability, right? So we are going to allow the passage of some molecules, but not others. And we will see examples of this as we continue here. Largely, this poses a barrier to hydrophilic molecules as well as different ions, because it's hard for a hydrophilic molecule to pass through this hydrophobic interior. Same with ions. Right? So this bilayer, it's important because it allows the cells to regulate what goes in and out of the cell. Now there's different types of membrane transport, right? So we can have a simple diffusion, which is where substances move across a concentration gradient, right? We see in our image here, we have a higher concentration on one side and a lower concentration on the other side. And we are moving across this concentration gradient from a high concentration to a low concentration. Now the same is true for facilitated diffusion, right? We're moving across a concentration gradient from high to low, right? But now in this case, we're aiding this transport with a protein, right? A transport protein. Right, and this is helpful, and we'll talk about this here, particularly for ions, right, and any hydrophilic molecules. Now these two types are passive transport, meaning they don't need any energy input in order for movement to occur. Right? They simply follow a concentration gradient. Now active transport, right, is completely the opposite. Right? First of all, we're moving substances against a concentration gradient, right? So we're moving substances from areas of low concentration to areas of high concentration, right? Molecules don't want to do this by themselves, right? So we need an input of energy to help the molecules go against this concentration gradient. All right, we're going to start by talking about simple diffusion. All right, so simple diffusion, we don't need a protein to help us cross the plasma membrane. And if we're talking about simple diffusion, there's many different molecules that can simply diffuse across the phospholipid bilayer without any help, right? But the rate at which they cross varies depending on its chemistry, right? So if we look at size, small molecules will diffuse faster than larger molecules. And we see that up here, we have these very small molecules that diffuse relatively fast. Molecules that are nonpolar will also diffuse faster, right? So remember, if we have a polarity, we're talking about some atom in our molecule has an electronegativity, right? I will point out here that water is the oddball here, right? Water will diffuse across our plasma membrane, right, without the help of a transport molecule or a transport protein. We'll talk about that on Friday. The charge is going to depend on how fast it moves across, right? So remember, polarity has to do with covalent bonds and electronegativities. Charges are really, we're talking about ions, right? And we can see examples of ions down here, right? Sodium, potassium, magnesium, right? They have a full charge to them. Now, if they don't have a charge, so non-charged molecules, they will diffuse faster than molecules with the charge. And finally, concentration, as we saw on the previous slides here, right? Molecules like to move from areas of a higher concentration to a lower concentration, right? And if that gradient is steeper, meaning it's much higher on one side than the other, that will impact the rate of diffusion. So if there's a higher concentration gradient, it's going to cause faster diffusion. Now, if we're talking about simple diffusion, right, again, we don't need a protein here, right? Particles are simply going to diffuse down their concentration gradient. Now, I like these images. So if we're focused on that top image for a second here, we can see we have a large concentration of molecules on one side versus none on the other side. That's going to drive diffusion across our membrane, right? But it's important to note that some molecules may not diffuse, right? They might simply stay on this side, right? As we get more molecules on the opposite side, maybe some molecules are going to start diffusing back across from where they originally came from. But the overall direction of diffusion is going to be with that concentration gradient. Now, the same applies in this picture down below, but this time we're talking about two different molecules, these green molecules and these purple molecules. Right? We see a concentration gradient exists. We have a higher concentration on one side for our green molecules, higher concentration on the other side for our purple molecules. Right? They are going to diffuse across the membrane depending on which way their concentration gradient is. So green is going to diffuse to the right, purple is going to diffuse to the left. Right? Now eventually in both of these situations we are going to get an equilibrium where there's equal amounts of our solutes on either side of our membrane. Now this doesn't mean that they stop moving in between the membrane. It just means that the movement that occurs is still in equilibrium, right? It's not moving more in one direction than the other, right? But it's relatively an equal amount of movement. We have reached equilibrium. Now, if we talk about facilitated diffusion, now we're talking about the use of transport proteins. And these transport proteins are super important for providing a passageway for ions and hydrophilic molecules, right? Because ions and hydrophilic molecules, right, they're very water soluble, but the interior of our phospholipid bilayer, right, is hydrophobic, right? And that doesn't mix well. So now we have these proteins that help our ions and our hydrophilic molecules cross into the cell or outside of the cell. Again, they're crossing based on their concentration gradients. Now we have two different types of transport proteins. We have channels and we have transporters. So if we're talking about channels, channels are simply this open passageway. It's a transmembrane protein, and it allows for the ions to pass through the phospholipid bilayer without having to interact with those phospholipids, particularly the hydrophobic interior of that bilayer. Now, oftentimes you might see simple protein channels that are always open, right? But most of the time they're going to be gated, right? And so the cell can have some sort of control as to when they might open the channels or close the channels. And again, we're talking about diffusion. It might be facilitated diffusion, meaning we have a protein, but that diffusion is still going to follow the concentration gradient. The other type of transport protein is a transporter. Right? They're also known as carriers. And these transporters, instead of being a constantly open channel or a channel that is gated, so it's open sometimes, right? These transporters will actually go through a conformational change to move a solute across a membrane, right? So the shape of this protein will change and move the solute across the membrane, right? Molecules such as sugars, amino acids, nucleotides, they will utilize these transporters. If we look a little bit closer at its structure, right, there's these really two important structural features, right? We have this hydrophilic pocket, right? It allows for solutes that are hydrophilic or ions to go inside of this pocket, right? So it's open to one side of our membrane. Once that binds to the pocket, right, that protein is going to change its shape and actually open towards the opposite side, hence releasing this solute to where it needs to go, right? So this protein has the ability to change its shape as it's working. Now there's many different types of transporter proteins. These are kind of the three most common ones here. Pretty simple to understand. Uniporter, so uni meaning one, it's going to transport a single solute moving in one direction. All right, so it's taking in a single solute, moving it to the other side. A symporter is able to transport two or more ions or molecules in the same direction. Right, so it's open to one side, it's taking in more than one molecule, making its conformational shape change, and then releasing both of those molecules to the opposite side. So they're still moving in the same direction. And then we have an antiporter, right? So in an antiporter, we're moving two or more ions, but we're moving them in opposite directions. And so in this example here, we have a solute coming in, right? And going into this pocket, we have a conformational shift and that solute leaves. But now, because it's on the open to the opposite side, a different solute is going to come in. And when the molecule changes to be open to the side it was originally on, it's going to release that solute, right? So two or more ions transported in opposite directions. All right, so far we've been talking about passive transport, does not need any import of energy. Let's transition to active transport. So active transport requires an input of energy because we're moving solutes against a gradient, right? Now solutes, they don't like to do this. It's really energetically unfavorable, right? And so to get solutes to move, we have to input a type of energy. And the energy that we're using here is going to be in the form of ATP. Now, two types of active transport that we will talk about. The first is primary active transport, and the second is secondary active transport. So let's focus on primary active transport first. So it's this lighter purple color here. So primary active transport is going to use this transmembrane protein. Right? It'll take in the solute, and in this case, the solute is a hydrogen ion. Right? It's going into that pocket. right? In order to get that conformational change and release this hydrogen ion to the opposite side, we have to input energy, right? So ATP is used, right? Now notice we are moving these hydrogen ions from a side that has relatively few to a side that has a lot, right? That's going against its gradient. Now at this point, we've built up this gradient, right, on this side of our membrane here. And so now we can talk about secondary active transport. So secondary active transport is using this gradient that was built up by primary active transport to facilitate the movement of yet another molecule. So in this example here, right, we have built up this hydrogen ion concentration to be really high on this side of our membrane, right? These hydrogen ions are going to bind to our transport protein, right? As well as our sucrose molecule is going to bind into our transport membrane, right? Now what's driving the movement here is not directly ATP, but it's the movement of this hydrogen ion from an area of high concentration to an area of low concentration. And in the process, it's taking a sucrose molecule along with it and moving this sucrose molecule against its gradient, right? So in this instance, active transport is not directly using ATP, right? But it's utilizing a gradient that was set up by the actions of primary active transport that did use ATP. So if we didn't have ATP at all, right, we would not be able to use secondary active transport. All right, so we went through a few different types of transport here. We went through simple diffusion. So in simple diffusion, we're moving from an area of high concentration to low concentration. Gases like to do this. Small hydrophobic molecules can easily diffuse. Water diffuses across a membrane via osmosis, which we will talk about on Friday. Okay. We also have facilitated diffusion, right? Still doesn't require any energy, but instead involves the use of a protein. Molecules that will move in this fashion will be hydrophilic molecules and ions. And lastly, we talked about active transport, right? The big thing here is that we're moving a substance against its concentration gradient. So anything that goes against its concentration gradient will be utilizing some type of energy, generally ATP, and going through active transport. All right, we will review this at the beginning of lecture on Friday, but that is all I have for now.